In this video, we will discuss about how the immune system function. More specifically, we will discuss about interaction between T cell and B cell and how they kill the infected cells. So let us start our discussion. So this is presented here is the antigen presenting immune cells, as we have discussed from the in our one of our previous videos, that dendritic cell when interact with the antigen of tumor cell, it initially eats up the tumor cell, and after some time it matures itself into the antigen presenting cell. And what happens when when dendritic cell matures into the antigen presenting cell, it interacts with the T cell. Specifically, there are two kind of interaction. One is the epitope interaction, and another one is the interaction to give a signal about the give a signal or information about the environment of the tumors. That So in this, the T cells that are interacting with the antigen presenting immune cells have CD4 receptor at the cell surface, and that interacts with the antigen presenting cell. At the same time, the second signal is of the T cell receptor interacts with the antigen presenting cells that is presenting the antigen of the tumor. So here this yellow sign is known as the epitope, which is the signature of the tumor. And so after that, the T cell receptors gets activated. T cell receptors get activated after it received the epitope signal. At the same time, the CD4 plus also interacted with the antigen presenting cell. And after the T cell gets activated, both of them releases cytokines, and these cytokines of T cells helps to replicate the T cells into, or we can say, this helps the T cells to make its clone. So here the clones of the T cells are formed, and this clone T cell then release cytokines that helps to activate B cells and CD4 plus cells. So B cells also get activated, and CD4, not CD4 plus, CD8 plus cells. CD8 plus cells also gets activated. And B cells, as you know from our one of our previous videos, B cells uh, works late compared to the T cell. T cell works faster. So now we understand from the sequence why T cell works fast and why B cell works or B cell activates later on. And now B cells works. B cells also has the information about the epitope of the tumor, and it works towards killing. It works towards killing the tumor cells. And the CD8 plus cells also, CD8 plus T cells also gets activated with the release of cytokines from the clone T cells, and it has the cytotoxicity that is injected into the tumor cells. So in this video we will discuss about CD8 plus T cells. So let us go to the next slide. So this is CD8 plus T cell. This is CD8 plus T cells that has cytotoxicity, and when it interacts with the infected T cells, how the cytotoxic T cells knows that the cells that uh, with which it is interacting is the infected cells with the help of epitope, because cytotoxic T cells has the information about the epitope of infected cells. So when it interact with the infected cell, it releases two kind of proteins, or we can say in general form. One is granzymes, and another one is perforins. So let us understand the function of perforins. What perforins does? Perforins makes the holes or makes the plasma membrane of the infected cells permeable, and so that these granzymes can penetrate inside the infected cells and can kill the protein or break the protein and lysate the cells. In this way, the infected cells get lysed, and, and this process continues till all the infected cells get uh, destroyed within the host. So this is how the normal immune system functions. More specifically, how the T cells and B cells functions, and why T cells activate first and B cell activates later on. So that's all for this video. Thank you for your time.